Good early evening, everybody, and many thanks for coming. My name is Professor Alex Leff. I'm one of the Deputy Directors here at UCL Queen Square Institute of Neurology. This is the inaugural lecture for Axel today, and I'm just going to do a very brief introduction. As is traditional, Axel will do his inaugural lecture, which is not like the standard lecture. I'm sure there'll be some science in there, but also a little bit personal as well. And then we have a vote of thanks from Sir John, and then I'll wrap things up. And there's drinks and nibbles afterwards. So looking through Axel's CV, I mean it's very impressive. There's a lot of double things. He's double trained as a neurologist and a neuro-ophthalmologist. He trained initially in Freiburg in Germany. I don't know if we'll hear a little bit about his German past. He then did a doctorate a couple of years later, and not happy with that, he then did a PhD after that. So I think he's Doctor, or Professor Doctor in Germany, but he doesn't like all the titles that come with it. He moved over to his clinical training at Queen Square in the late 90s, which is in fact where we met. I remember us meeting in ITU over some interesting patients, I think with some autonomic problems. So we've sort of had parallel careers, and we've been following each other's careers for quite a long time. Axel is an incredibly busy clinician. He's got a dual appointment here at the National Hospital, but also at Moorfields. And has a variety of clinics in his name, seeing over 2,000 patients a year, which is really very impressive. He somehow, despite this busy clinical time, he finds time for research. He's published over 300 or nearly 350 papers, over 100 of them as first author. He's brought in over £6 million worth of grants while he's been in the UK. He's fluent in 5 languages. It's all rather annoying, isn't it? Anyway, I think I've said enough there. He's an amazing clinician and academic. Some great clinicians that we've worked with in the past. Certainly, one of them said to me that if a good neurologist must know their neuro-ophthalmology, and if you know your neuro-ophthalmology, you know your neurology. So, with no further ado, if we can thank Alex as he comes to the lectern to give his inaugural lecture.
Thank you for the kind words, and thank you all for coming to celebrate today with my wife Laura and our 3 children, Elena, Max, and Kasper, who's 4-year-old, that the eye really is an open window to the brain. But in order to see through this open window to the brain, you really do need to have a main actor. That main actor is a small worm, and she is called Phila. Phila does live on a little asteroid, and this asteroid every now and then passes planet Earth and has been looking down to what's happening at Queen Square for quite a long time. Only very few people sitting over here know what all that was about 7 years before it started. I'm ever so grateful, Lady Dean, Sir John, that you come together with your children to actually celebrate the end of this journey, or maybe it is the beginning. But it is a custom of this series of lectures that the audience has to learn something about the speaker. Now, it would be completely inappropriate for me to say something about myself because this would be entirely biased. Therefore, I retort to those people who know better, and who better than your teachers in school. The lecture will work along titles. The first title is The Bumpy Road. That's the beginning, chapter 1, The Bumpy Road. And what you see here is my first assessment in school in year 1, making the transition to year 2. I went to school in Heidelberg. It was the International Unifying School. It was a school of peace and for peace. And I think it is part of this peace business that notes which were given only were permitted to be very good or good. And on that German system, if you didn't do so good, then you probably got a stamp in your assessment which says you participated in English. That was a bumpy road, and you can see little feeler looking here where this will go. Now, I just want to point out one presentation or one publication at the very top of this slide, because I will come back to it at the end. And that was work done here in the intensive care unit with many, many people. I won't call out all of the names, but I'm grateful for where we looked into the brain of people with traumatic brain injury and just collected the fluid from around the brain to examine what that could all imply. We couldn't explain it all at the time. But back to chapter 1, the bumpy road. The bumpy road. Chapter 1 is the only chapter which comes twice, because the principle of science, or the mother of science, is reproducibility. And what would you believe if only one teacher who may not have liked me said in primary school that I'm unable to speak English? What you see here is reproducibility over the course of 13 years by 3 consecutive generations of teachers. The German system goes 1, 2, 3, 4, and 4 does mean that you are actually about to fail. 5 is fail. And on the left, it is written ausreichend, which means it is a 4. Then to the top, about 3 years later, you see that they are actually fighting with the fact. They want to give me a signal. 4 minus doesn't exist. 4 to 5 doesn't exist. And I think the message was really stay a 4, because otherwise we kick you out of school, which I managed to do the subsequent year. But only to read this out to you, it says, it translates from the left in English. He does make some efforts, but has great difficulties, particularly expressing himself in spoken language. So good luck with the next 34 chapters. Chapter 2, the mysterious eye puzzle. If it wouldn't have been for Goulden, I would not have been at Queen Square. There's no doubt about it. And what Goulden has done, he said, over all these years, put puzzles in front of me, and I can see Niels is sitting here, and Alex referred to the fact that we had been in clinic. We had been learning from Goulden all these years by his ability actually to let us think about these puzzles having been put in front of us. In the clinic I just came, still has the same problems, droopy eyelids, coordination between eye movements and hand movements, and all that. But there are 2 more mentors to mention. The first is Ted. Ted did teach me all about color vision, color memory. And the first paper up here on new memory in young children was inspired by Darwin and his thought that his children would be colorblind. Of course, it was unfounded. And the research we did show it. But we found out that color vision developed at the age of 4 to 5. And I was pretty proud. I got a doctorate for it. But Lena, when you were born, sitting in front of my bicycle and at the age of 2 years could call out all the names of all the colors in the cars, I somehow knew that science is. either children change or you're particularly able to see colors, but it was very different to what I had found. Now then there is Ed Thompson, and Ed has an ability beyond anything. I think Nick Davies, we can agree, the intellectual freedom we had in his lab was unparalleled. He did let us develop our own ideas, and I will be eternally grateful for that. Chapter 3, a thirsty worm. It had been a bumpy road, it had been a long road, so Fila needs something to drink. But Caspar, what should she do? Should she drink from this green, muddy puddle which is smelly, or should she drink from this clear, crystal clear water? Exactly. So the message really to the audience is you would choose the water, but as always, it's the dose which makes if something is bad or not. So too much water is no good. Important for school holidays or Duke of... How's the Duke called? Exactly, challenges. For those who run marathons, and I look over in this direction of the audience, be careful with your rehydration. And then to any junior doctors present in this room, if ever someone puts a bag with glucose in your hand, think twice. Put it away, take normal saline, because that is what keeps your patient alive. And finally, you may have read in the notes, There are forms of alcohol which are not too good to touch, taste. Methanol definitely shuts for good the eye as a window to the brain. Chapter 4. The yellow glow detective glasses. That is a part of research which kept me busy for a better part of 10 years and had to do with the fact that if you bleed into your brain and that is missed and you bleed again, you have a chance of 95% to die. But there is the possibility detected with a very, very elegant method which was really pioneered in the United Kingdom at the time. Here you see Felgner taking on these special glasses, and in which you see in the middle in yellow these type of patterns emerging. That is science. You can plot it in X and Y coordinate, but really all you want to discover is a little bit of yellow, either in a melange of reds or whites, and that is not possible other than by using this method. And it has been over the years a pleasure to work together with the group in Sweden. They now do it like this. Of course, with my German colleagues, we have very, very good relationship. And they do it also the way it is done in the United Kingdom. Chapter 5. The mirror that sees inside. Somehow we need to get this window. Somehow we need to look through the eye into the brain. And that is Felgner doing her maquillage in front of the mirror. And she discovers that if she sees the depth of her eyes on the other side, she can see her brain. And in my medical career, that was one technique which was invented, went along, and a series of very, very productive PhD students, starting with Preyas, Tevari, Lisanne Balk, then we had also Jenny, we had Danko Courage, and Emma, you are here, you're bringing that to the next level, are looking at these nerves and have really built on the clinical experience, made that this is of good quality, that it develops further, and that it is related to disease and helps patients with their diseases and the management of the diseases. Chapter 6. The fiery flame behind the eye. So as Felgner looks into these mirror, she feels like a fire burning behind the eyes, and as she moves these eyes, this is unpleasant, her vision dims, and that is what has become the disease I'm most involved with, optic neuritis in all its different forms, coming back and eventually causing patients, this is Neil Wiegering, who reported us, once you have the diagnosis and you know it can come back, it is like a Damocles sword over your head and you're very, very worried. So it is important to be able to tell patients if they have a condition which returns or if everything is settled. Chapter 8. The fraying retinal threads. So what will you see if you look through this mirror inside yourself? Using analogies like spiderwebs. If you look at your windows in the winter when you haven't cleaned them for a while and there's a spiderweb before it, you see all the fine patterns in it. Then you go to Newcastle, it will be the same pattern of spider nets. You go to Liverpool, no change. Then you think maybe it's different if we go over to the French, same pattern. You go to Australia, same pattern. And that is what we have done together with a group of very, very experienced spiderweb watchers in the Lens Generation in 2010, and we thought, well, maybe these spiderweb change over a 10-year period, but it is absolutely reproducible. So of all those tests I've seen in my lifetime in medicine, these spiderwebs are absolutely reproducible to the decimal point. And that is why very early on Danko Courage discovered that we can use them to diagnose these problems, because they always reproduce. And that has been so successful that it's now included in a number of diagnostic criteria and upcoming diagnostic criteria. Chapter 9. The vanishing nerve sparks. Nick, this chapter is for you. It has been the time in your department looking at Guillain-Barré and having these discussions where we found that as Felgner disintegrated, could not wiggle her tail, couldn't feel, You think she will die. She looks like she's dying on this slide. But in reality, it's a disease called Guillain-Barré syndrome, which makes almost always a full recovery. And it has been really your guidance, which took me to the point to understand how feeler, if we measure it, does actually permit us to say when it may not be all that good. And that is something which has over the years been taken on by other groups. And Mike, I'm very, very happy to see you here and people from the Department of the Neurology Lab, because your group just delivered very recently the smallest sibling to feeler called peripherin. Chapter 10. The twin signals of glia and axon. These are the terrible twins. You see how feeler is actually shocked by them. And the problem is, whatever you do in neurology, you cannot have only the axon or only the glia. They always come together. They develop together, they stimulate each other, they repair each other. But most of all, remember, feeler is still dreaming on this asteroid going around Earth. There is this nightmarish chapter 11 in the trembling threat signals where Earth shakes and she is afraid it would break apart and everything come out. And that is one of the diagnoses I do actually still to date not like to communicate to patient, because to me it feels like a death sentence. That is motor neuron disease. In motor neuron disease, you have so much of these filaments being released into the bloodstream that you know people don't have very long to live. And it has been one of the nicest moments in my career to see that all the work we have put into developing these tests, validating them, coming up with a roadmap how they should be included in trials a few years ago led actually to the first treatment in, I think, over 50 years to be used in patients with SOD minus ALS. And it has been getting rapid accreditation and approval by the FDA and the EMA solely based, and this is really, really important, solely based on the results of neurofilament levels. So that now has become something where patients have real benefit. Chapter 12, the tangled threads. Chapter 12 could also have been called Chapter Pretzel, but it would have been too German. So as feeler knots herself up and you try to pull to untangle her, it leaves, of course, scars and things which you will see in the future. You can predict 15 years ahead that people will have some form of brain rot or shrinkage, that diseases will do worse. But one of all these studies is closest to my heart, this in Annals of Neurology, entitled Evidence for Acute Neurotoxic Effect After Chemotherapy. That was a trial at the time run in the Netherlands by a friend of mine, Rogier Hinsen, who looked at people with multiple sclerosis, gave them strong chemotherapy to replace the immune system. The trial was negative, and he gave me all the blood samples and Axel, look at neurofilaments. Maybe you can find something. And the hope is always, of course, you want to do something well. So it was not a very pleasant moment when I took the phone and said, Rogier, I think you have done something, or that intervention had done something really bad to your patients, because after they got that, neurofilament levels by 10 times went up. They all lost the ability to walk. So we think what happened is that because the blood-brain barrier, and I'm looking up in your direction, is short, all these chemotoxic agents come through and just destroy the neurons. And the discussion of this paper is worth reading because it sets out why neurofilaments should be included in clinical trials as a safety measure. And many of us now also at Queen Square have had that experience that trials are closed if neurofilaments go up, even in otherwise rapidly neurodegenerative diseases. So that is a key point here. Chapter 13, the broken backbone bridge. This slide is only here to remind you to stretch yourself for posture. Chapter 19, chapter 14, the shaking steps. it would be impossible to do anything in neurology without speaking about Dr. Parkinson and all these movement disorders, which have been part of the foundation of this place. It also tells you if you do neuroophthalmology, you actually get in contact with a lot of different disciplines. It's almost impossible not to speak to peripheral nerve neurologists, to speak to people who do epilepsy, surgery, ophthalmology, and so also in the Parkinsonian syndromes. From my perspective was interesting because early on we discovered these proteins to be released, and we know they aggregate. But with the more recent studies of the mirrors as a window to the brain, optical coherence tomography, the group of Siegfried Wagner and Piers Keen found out that a very specific part is affected. So if you look at a spider web, and you would always find that the fourth inner circle gets a funny appearance, then you would wonder what is going on there. And this happens in no other disease. And we do think this is interesting because that is where you have alpha-synuclein and proteins like this forming aggregation. They're toxic. So this would be the first, really, it would be the first model where we can look live into the brain, how degeneration possibly develops in related to a real protein problem. And of course, if you prevent it, can also have ways of how to make this, to reverse that if possible, because aggregates you can resolve. Chapter 15, the cave of fading patterns. Max, imagine you go into a cave where you see all these hieroglyphs. And then as you look at them and want to find the meaning, they disappear. That is how, for many patients who have a form of dementia, posterior cortical atrophy, it feels. They see the patterns, they don't put them together. And working together with the dementia group, and Nick Fox also came briefly before, they have a meeting today, and particularly with Kier, we developed this test, which is called the Graded Incomplete Letter Test. And that is another strength of working the United Kingdom, because that got integrated into United Kingdom Biobank, which is 500,000 of people, and that is the world's largest cohort ever being tested for impaired higher cognitive function. We have all the genetic, we have all the brain imaging, we have all the imaging of the eyes. So over these years, Alex, if you ask me what are you doing, this will be one focus where there will be more productivity, and it also involves passing it on to the next generation of researchers. Chapter 16, the lost memory game. If I play memory with my children, Lena always wins. Kasper comes second, and Max and Laura, thank you for letting us play. It's not without participating all the time. So you can see here Fila losing memory cards with this other worm, so she only got one set correct. And that is what happens in dementia. You just lose your memory very, very slowly. And when I came to work in Queen Square, essentially the cerebrospinal fluid was meant to be normal. So you wanted a clear water fluid, nothing in. And that fundamentally changed over the last 20 years, so that now we use all these proteins in the cerebrospinal fluid to tell us what type of dementia it even is, if it responds to treatment, hopefully. And I would not be surprised if these treatments work, if Queen Square could get another Nobel Prize, because that is where the disease was discovered. But another problem with memory is, and dementia, that there's so many forms of it. It's almost like a maze. You remember the stories of the hero walking into the maze, fighting a monster and not finding the way out. And then he got a threat to find his way out. Fila has it easy. She goes into this maze and her body is a threat. And that is what makes proteins very attractive. And Mike, your lab just got this new tau 217 phosphor. So on proteins, you get an encoding of what happened. And you can just roll the thread back, and it tells you a lot about even very, very slow diseases. Then there are, of course, those people who are obsessed with detail. Chapter 18. The whispering retina rulers. In short, you just have to accept that whatever you do, you have about 2% of variability anyway. You measure your height in the morning, a bit more than in the evening. What a surprise, no different for proteins or for looking at the back of the eye. If you know it, you can just control for it and repeat things as much as possible. But there is a particular thank you to the editor of Brain who permitted me to write a scientific commentary. It's like an editorial, it's something no one reads. And that keeps tracking citations because this retinal lymphatic system wasn't even proven experimentally by the time it was discussed, and now we have it. That is, if you want, the way how the garbage will be taken out of your eye and your brain. So I'm very, very grateful, Dimitri, for this. Then there is the next chapter on the magical severity scrolls. It's a bit of an annoying chapter because whatever you do, it needs to match what is written. So in medicine, we have all these numbers to rate how patients do. Patients sometimes say it is not exactly matching it. And what a surprise, look at the facial expression of Felgner at these two blotchy scrolls. There will never be a perfect match. And most of the things we do are complementary. Most of the things we do at Friends are complementary. Caspar, can you imagine by night going into the kitchen and doing mischief because you want to have some sweets or a cake? The tiny invader mischief. Look how Felgner goes over the birthday cake before it should be eaten. These tiny invaders in neurology are the infectious diseases we see, and many of the infectious diseases come along and they are a hype for a certain period. We all went through one specific period, but long before that we had West Nile virus infection affecting the Americans. And what we have seen in COVID is that this pattern, how it looks in post-mortem, how it looks in the blood and so forth, very much repeats itself. And I'm grateful for work with particularly Johann Ehler on looking at larger cohorts, also internationally, of these patients, and then finding out that if you have COVID and you're very sick, it does not make you so different if you have another bad lung disease or sepsis and are very sick. And I think the message here is really a positive one to us as neurologists. If we see sick patients, as they are very sick and very vulnerable, they accumulate nerve damage. We just need to be aware of it, independent to whether this is COVID or something entirely. But I can feel the next chapter, the whispering gas bubbles. You are now looking forward to have a drink, and I can promise you none of the nasty substances listed here to the right will be included. Chapter 22, the river of red drops. That is the consequence of all forms of bleeds to your brain. Felgner just swims in it, and it is exasperating, and we haven't really achieved very, very much about it. But these types of proteins permit very early on to discover some of the type of secondary problems people may develop. Moving from the red into complete different color, matching the chairs in the room, the aqua antibody waterfall. We looked at all these patients with optic neuritis here for probably 50 years. They were passed on from generation to generation. Ian MacDonald, Gordon Plant, and there are many of us doing it now. Then these antibodies were discovered, and suddenly we could explain it. We could tell patients why do they get it again, having the antibody or not. They are then the mock antibodies. They are done all here at Queen Square in the department. Then there are a number of other antibodies, autoimmune encephalitis and all that. And we can treat them because we need to suppress the immune system. There is epilepsies. There are seizures that respond to steroids. Something which I've been heresy saying before in this place. The perfect testing potion. The perfect test doesn't exist. If we validate this, we send it around the world, Canada, South America, Australia, everyone gets a different result. even if we standardize it, automate it, it is very, very difficult to get a perfect testing potion at all. And even if you think we are there at the moment, then one of the things I do as a side hobby in the evening is looking at some mathematical ways of showing that maybe there is more to be done to make that really individual and reproducible. Chapter 25, the great CSF treasure. This is from my time as a pirate, Casper, where you got a treasure chest. And it was the treasure chest of Ed Thompson, which I did steal there. So he did put everything into the cerebrospinal fluid and said there is everything we can learn about the brain in there. And that is something which I carried with me ever since, but working together with different people in different countries, because we do need to have a consensus. How to do things, we need to do things in the same way, and sometimes also very small observations on that make it in subsequent revisions into diagnostic criteria, in this case multiple sclerosis. Chapter 26, the midnight brain map. If you're a doctor, you have to do these night shifts, and a lot of things at night you encounter, you have to deal with. And the one thing to remember is there will never be one single drug which solves the issue to you. What you really need to have is good nurses and a good team. We have a workforce which does need more moral. We need more people, but the nurses we have, they are there really to keep these patients in difficult situations alive, working, and happy. And that is the best, whatever the situation is we have. So no drug will ever solve that. And you need to stand up if the phone rings. This is the only chapter where Fehler is on the other side. The ancient brain stones. That chapter is the one where I thought for a long time this would be a career-ending move, because I got hold of an ancient brain from Haslington, 2,600 years, which I sort of munched up, investigated over a period of 10 years, and then could show that Fehler, just as she crawls out of this cave, does exist that long. Absolute career-ending move. And as it happens, Fehler discovered in this cave other things. There's a book of Charles Dickens, that's Bleak House, where he describes optic neuritis better than any doctor. And Charles Dickens had described a lot of different diseases, and you know he attended all the post-mortems of the then founding editor of The Lancet. That was the spirit. Chapter 28, the virtual neuro journey. This and the following slide are completely random. In this slide, there is everything which is simulation of stoichiometry, mixtures, thoughts about how neurofilaments can be used to introduce genes to do neuroprotection, going in the next chapter, the scrolls of tiny discoveries, to themes like astronauts. I would never have dreamt to publish about astronauts, but you need to respond to letters you get from people. And all of this will merely serve as a footnote in science, but that is important. Chapter 30, the quest for healing clues. And you can see Fehler is trying to catch this mouse, and this mouse is running away quick. The main message in neurology is be quick. If something happens, don't wait 3 months for someone to come in and then try to run after and say, I'm sorry. It doesn't matter if it is in multiple sclerosis to prevent axonal damage. Definitely in optic neuritis, at the onset you can treat patients, and if you can stop inflammation before it has done too much damage. And Sally, in our clinic, this is probably one of the main things we're able to deliver. Sally and I do a job share, but more than that, we actually share believing that we can prevent vision loss in these patients. But to do so, there's only 2 of us, and we can't do it alone. We need to have support. Chapter 31, the world of young healers. That is something which Rob Haddon, Konrad Radak, Albrecht Günther, he's on here, who I saw last week in New Critical Meeting in Germany, set up together it is still something where I'm most proud of, the young neurology trainees. At the time, there were big conferences, professors meeting each other, and the trainees, we felt intimidated, and we couldn't pay for it anyway. So you will now see that at most of the meetings, there will be young neurologist sessions, there will be prizes, there will be travel grants. So all of that originates in a time where we set out with a lot of energy to network that. And these networks are still the networks I live today from and I very, very much enjoy. And this lower part where the Lancet Neurology got engaged with is also something I will be involved in the years to come, because we have a specific idea about this training in optic neuritis, which is ongoing as we speak. The firestorm potion. Well, there are parts of medicine where you just feel guilty. And if you look at embolic visual loss, so you get an embolus in a central retinal artery or in a branch retinal artery, you get blind. We have not done anything in the last 150 years about it. We are still completely unsuccessful. We have about an hour to do something. We send patients over to Moorfields. They sit there for hours before they're seen by a nurse and then told to come back the next day. And that is where I'm very grateful of the close collaboration with the stroke team at Queen Square, particularly Arvind, that has taken interest in treating these patients rapidly. And I hope that before I retire, we will take acute visual loss just as serious as we have taken stroke, because that has been very, very successful in stroke period, to be quick. Chapter 33, the soothing moment. We are almost there. Caspar, the ice cream is on the screen. Anything happens in life, a bit of ice cream definitely does help. There is a lot of pain in the eyes, and Emma, this is the last part where you will be working on the tiniest of all nerves. These are the C fibers, the nerves we cannot really see anywhere, we cannot measure anywhere, and you will look into what that means in disease and what we can do with it. If we can influence it and can make things better. We're coming close to the end. You can see it has been a bumpy road to start with. It has been a long road. There has been thirst on the road. And here, Fila approaches all these friends, waving with the flags. So she hopes there's a bit happy ending. I'm not sure about that. But the message from this is all this type of research only ever could develop something by uniting people, doing it together, spreading the word, and having more and more people engaging on it. We have quality control criteria, we have a postal recommendation, and that is something which is so, so important in this time for artificial intelligence, because this is what will develop and deliver the high quality data we desperately need to save this health system and to make use of the electronic help we do have. Almost finished. This is chapter 35. Remember, Fila was on an asteroid going around Earth, and this asteroid got off course and is now smashing into Earth. Before it will do so, I just want to share that, of course, optic neuritis has been one of my main interests, because that is where the window looks to the eye. But it has been an incredibly gratifying journey to work with over 100 experts from over 100 countries around the world to find a global consensus on how to investigate that condition, how to diagnose it, how to classify it. And there's a big thank you also to UCL Library, which helped to translate that in 114 languages and made it available for free for download. That will be the part, Alex, where most of my work will rest in the last 10 years of my career to come. But you can feel it coming. The last chapter, the perfect protein potion. Fila did not make it to survive. As she approached Earth, she smashed into a silver sieve. And not like a spaghetti moving through the thread. She just hit it like that and fell into pieces. You remember the first slide I said, I will come back to it. We implanted these little sieves into people's brains. At the time we couldn't explain how such a big worm like feeler ever would go through the sieves. We had some reason to show that it was smashed up, but in this paper, which is a personal hobby in the evening, it was possible to show that you end up with 1,400,000 odd fragments of this protein, which you can very nice catalog, and it's something which you can transmit to all of the other proteins, and that is what will enable to move from these expensive tests we have at the moment to more high throughput simple tests which are available all around the world. But again, you have seen the progress. It takes about 20 years to be there. So that is for other people to do. And with that, I really do want to thank my sister, Barbara Petzold, who has sketched all the images of feeler. She's still on her asteroid. She's waving over to John and says, I'm passing the stick on. Thank you.
Well, how to follow that? That was very, very impressive. It's a bit daunting for me. I'm just a BA history, so I'm rather out of my depth in such a high-powered medical and scientific environment as this. But you'll be relieved to hear that I'm not going to give you my considered and profound scientific views about Professor Petzold, the scientist. I'm going to tell you a few things about Axel, the man. Axel came into our lives, I can't remember the exact year, but he came into our children's life when he was in Colombia. And I have to tell this story because it's really encouraging for everybody else, because it shows that you don't have to start well in life on the medical side in order to produce a good result in the end. And the story is this: Axel in Colombia sees a coconut tree, decides it would be nice to climb up the coconut tree to give his friends coconuts. Gets up to the top of the coconut tree, gets the coconuts, hands them down. Thinks it was quite hard climbing up, but I can climb down very easily. So he slides down the coconut tree. Now, as you know, coconut trees are not structured to be slid down. So he arrives at the bottom with his legs quite seriously lacerated. And because at that stage he wasn't yet a professor and he didn't have any medical or neurological qualifications whatsoever, he paid no attention to getting it treated. He just lay around on the beach in the sun, got lots of sand in it and dirt in it, and within quite a short time he was pretty unwell. The moral of the story is you can be at the bottom of the tree lacerated with a very badly decomposing leg in Colombia and still become a professor in your later years, let's say. Diane and I first met Axel when, I think it must have been the late 80s. No, it must have been the late 90s, actually, the 90s, when he was a very young man doing obscure work at a place called Queen Square that we'd never heard of. Friends of his told us that he was following some research leads that could be very productive, they said. Could be very productive or not. What was obvious at that stage was that he was working very hard and very purposefully, and he was facing rather a lot of obstacles, not least funding problems, which I suppose are familiar to all of you. But behind Axel's genial, smiling manner, some of you may have noticed that there is some granite. I didn't realise that at first. I thought he was just a jolly German guy, you know, good jokes and all that. But we decided there was another side to this guy when our children brought him to our house in Brussels and we observed his approach to table tennis. We played quite a lot of table tennis in our garage. Now, I can't claim that Axel is the best table tennis player I've ever met. Probably one of the worst, actually. But one characteristic of his table tennis playing was that he was incredibly competitive and very committed and determined to win. And I think that has probably been one of the keys to his career, because without the energy and the commitment, I don't think he'd have got through the early years when funding vanished and he was living an extremely austere life as a poor postdoctoral research student. Sometimes he struck us as being a bit like the Ancient Mariner. You probably know the poem by Coleridge. And he would stop anybody he came across and tell them at great length what he was doing and why it was going to be very important if it worked. I didn't understand a word of the science, but it struck me that this guy is really pretty determined. He's going to go somewhere. Well, watching Axel was for us an insight into a broader question I wanted to talk about for about 1 minute, about how hard it is in the early career for young scientists and how much moral fibre they need to press on with a very uncertain project which may have a positive outcome but may lead absolutely nowhere. And that's something that doesn't happen on my side of the ledger in the arts, because you can always bluff your way into something. But in science you can often come against a complete brick wall and that's it. 2 years wasted. But Axel rather has his conviction and his hard work has been vindicated, and it's vindicated tonight in this wonderful account of his journey and now the coronation of a professorship. It's a good example to the younger generation when you think about his very poor medical start in Columbia. But it's also good news for the rest of us, we non-scientists who benefit in health terms from the results. Our family have very deep pride in what Axel has achieved. We're also very grateful for his friendship, for fitting so very well into this country, and for bringing into it the lovely Laura and the children. As someone whose family has actually benefited enormously from the work of Queen Square, I want to end on a personal note and just say how grateful the rest of us and our family are to all of you, younger and older, for the dedication with which you work and for the excellent results from which we all benefit. Thank you very much.
Well, thank you very much, Sir John, for a really warm and rounded view of Axel, giving us some extra bits of knowledge. And I'll watch out for those coconut trees. So we've now got drinks and snacks just back up the way you came. And Axel, I think things have gone incredibly well, and you've rather played within yourself, but it's nice the way you showed off all your papers. But if things do go wrong, you can always write children's books, I think, with your sister. And I mean, the Adventures of Feeler, I can see it now, lots of merchandise. I think you're onto something there. So if you'll just join me one last time and thank Axel, and please do stay for drinks.